Good morning, distinguished guests, speakers, ladies and gentlemen. A very warm welcome to the academic activity with Prince Mahidol Award 2022 Laureates on the theme of the HPV Day. This academic section is co-organized by the Virology Association Thailand, Department of Obstetrics and Gynecology, Department of Pediatrics, Department of Microbiology and Research Department, Faculty of Medicine, Laureates Hospital. I'm Bhakwar from Virology Association Thailand and Department of Immunology, Faculty of Medicine, Laureates Hospital.
And I am Radikon Akharawongsapat from the Virology Association Thailand and Department of Microbiology, of Faculty of Science, Mahidol University. It is our great honor to be MCs for today's academic session. This academic session is a part of Prince Mahidol Award ceremony, and of course we will learn more about human papillomavirus and HPV vaccine from our eminent speakers.
Yeah, we would like to extend our warm welcome to audience online. And if you would like to ask the question, you can type in the chat box. And we will try our best to deliver your question to speakers. Ladies and gentlemen, without further ado, may I please invite Professor Piraipan Putthawattana, President of Virology Association Thailand on stage to deliver her welcome and opening remarks. Professor Piraipan, please.
Honorary guests, ladies and gentlemen, on behalf of the Virology Association Thailand, the Department of Microbiology, Department of Pediatrics, Department of Obstetrics and Gynecology, and Office for Research, Faculty of Medicine, Laureates Hospital, Mahidol University, I would like to express my warm welcome to our guests of honors, distinguished Thai speakers, and all of you who join the academic meeting today. So, this important meeting is deal with human papillomavirus vaccine. Cervical cancer is an important disease burden for women worldwide. The WHO reported over 600,000 of cervical cancer new cases and over 300,000 deaths in 2020. About 90% of them live in developing countries. For Thailand, in the year 2021, we have over 9,000 new cases and higher than 4,700 deaths. So it's a big problem for women worldwide. The achievement of Dr. Lowy, Dr. Schiller, and Dr. Fraser in the discovery of a virus-like particle, which is derived from capsid protein of human papillomavirus, is a great innovation step that led to the HIV vaccine production. Unfortunately, Dr. Fraser cannot join us today. The HPV vaccine is highly effective. It is immunogenic and can prevent HIV diseases, cervical cancer, and other HPV disease and HIV-related cancer. It has been implemented for people, particularly women worldwide. So in the overall, the vaccine has saved enormous number of lives. In this special session, our guests of honor, clinicians an authorized person from the Ministry of Public Health will share their experience and their ideas and preventive approach against the HPV diseases. Finally, I wish the HPV Day today a successful, lively, and knowledgeable session for us all, not only for you who are in this room, but for the audience, a few more hundred who are online. Thank you.
Thank you very much ค่ะ Professor พิไลพันธ์พุทธวัฒนะ President of Virology Association Thailand for the opening speech. Now the academic session with Prince Mahidol Award 2022 laureates under the theme The HPV Day has officially begun. Now, next, I would like to introduce our first speaker, Assistant Professor Pathama Chowpothong. She received her medical degree, Diploma of Thai Board of Obstetrics and Gynecology, and Diploma of Thai Board of Gynecology Oncology, all from Faculty of Medicine, Laureates Hospital, Mahidol University. Currently, she is Assistant Professor at Department of Obstetrics and Gynecology, Faculty of Medicine, Laureates Hospital, Mahidol University. Her research expertise is in the areas of cervical cytology, cervical cancer, and AI colposcopy. Today, she will give a talk on overview of HPV-related cancers in Thailand, the burden and preventive programs. Please welcome Assistant Professor Pathama.
Good morning, Chairman, Co-Chairman, ladies, gentlemen, and all the attendees. My name is Pathama Chowpothong from Gynecology Oncology Division, Department of Obstetrics and Gynecology, Faculty of Medicine, Laureates Hospital. Today, I will talk about overview of HPV-related cancer in Thailand, the burden and preventive programs. First of all, welcome to Thailand, Professor. I know that there's very long distance from Maryland to Thailand. It's about 13,000 kilometers. For Thailand populations, last 2 years, we have about 66 million population. And 3/5 of them are between 25 to 64 years. This is the recommended age for cancer screening, especially in women for cervical cancer. About HPV or human papilloma virus related cancer, as we know that HPV can cause several cancer, for example, cervical, vaginal, anal, penile, vulva, and oral cancer. For oral cancer, this consists of oropharyngeal, oral cavity, and laryngeal cancers. Data from CDC last year show that cervical cancer is the most common HPV-related cancer among women, and oropharyngeal cancers are the most common among men. For Thailand, about HPV-related cancer in women, we found that cervical cancer is the most common, with the incidence rate about 25.6, and oral cavity cancer is the most common among men, with incidence about 7.87. And the burden of cervical cancer in Thai female is standardized incidence rate of 2 years apart, about 16.4, with the mortality about 7.45. However, the trend of cervical cancer in Thailand is slightly decreased in the last 20 years. I think because of in Thailand we have screening coverage. In 1990, cervical cancer is the most common in female Thai. However, in 2014, cervical cancer is the third most common, followed by breast, liver, and bile duct cancer, and cervical cancer. This slide shows the most frequent oncogenic HPV that found in Thai women. Most most of oncogenic HPV related to normal cytology to cancer. The most common are 16, 18, 52, and 58, as same as other countries. 67.6% of cervical cancer attribute to HPV 16 and 18. Because I'm a gynecology oncologist, today I will emphasize on cervical cancer, which we call preventable cancer, because we have primary prevention, secondary prevention, and tertiary prevention. For primary prevention, we have HPV vaccine. The years of introduced in EPI program is 2017. We give HPV vaccine to the 5th grade school girls. Now the percentage of coverage for 2 doses is about 66% in 2019. And because of COVID outbreak, last 2 years we lack of vaccine. However, last year we resumed the vaccine by quadrivalent vaccine already. This is a clinical practice guideline by Royal Thai College of Obstetrics and Gynecology for primary prevention of cervical cancer. The target group of Thai women are girls about 11 to 12 years, but the other group include female and male 9 to 26 years, immunocompromised host, and who with history of abnormal pap smear, abnormal HPV test, sin, and genital warts. However, for the other group, it's not covered in EPI program, and they should to pay by themselves. In 2020, Ministry of Public Health have the study about single-dose HPV vaccine effectiveness, and the study was in Udon Thani and Buriram province, and now we wait for the result. About secondary prevention, we have cervical cancer screening. The data from 2021 show that 60.2% of women aged between 30 to 59 years receive cervical cancer screening. We have 3 phases of cervical cancer screening. The phase 1, we use pap smear, and the phase 2, we use dual track strategy. That means pap smear and VIA. And the phase 3, we use HPV DNA testing. For the first phase, we use pap smear or cervical histology. Thailand used this strategy since year 2005 to present. We screen in the women 35 to 60 years with the interval every 5 years. The second phase, we call the dual track strategy. That means pap smear and VIA. In women 30 to 60 years, we use pap smear. But in women 35 to 45 years, we use VIA. Both techniques, with skin interval every 5 years. From this strategy, we can cover the total province of Thailand. And now we are in the third phase that used HPV DNA testing. Thailand used this method since 2020. And the female aged between 30 to 60 years are the target, and with screening interval every 5 years. Now the number of provinces that use HPV DNA test is about 60 from 77 provinces. And last year, Thailand initiated HPV cell sampling. About tertiary prevention of cervical cancer or treatment and surveillance. Now we have nearly 400 gynecology oncologists in Thailand. We have 1 National Cancer Institute, 7 cancer hospitals, 26 tertiary care hospitals, 25 medical schools. And I think I will finish earlier because I know all of you wait for the next session. This is my last slide. WHO announced the 90, 70, and 90 target. In 2018, that if you can follow this target, the cancer will decrease to less than 4 per 100,000 women. The first 90 means 90% of girls fully vaccinated with HPV vaccine before 15 age. 70% of women are screened with a high performance before age 35 to 45 years. And the last 90 mean 90% of women identified with cervical lesion or disease are receive treatment. Okay, thank you very much.
Thank you very much, Dr. Pathama. I think we have time for maybe a couple of questions. Anyone in the floor would like to ask question? Also, with the audience online, you can type your question in the chat box, and I will deliver the question for you. Yes, please.
This is Doug Lowy from the United States. That was a beautiful overview. I'm very impressed by how your cervical cancer screening program has been associated with basically cutting in half the incidence of cervical cancer. But I am surprised that your coverage of cervical cancer screening is so low. And the reason I say that is you would imagine that if you were only screening 2/3 of the people who were at risk, that you would not have a decrease as much as you have. Do you have any explanation of why the screening has been so effective?
In my field, the screening coverage in Thailand is about 60%. That is quite low than expected. Even we have three modality of treatment, three modality of screening. I think because the nature of Thai women usually are so shy to get an examination, and the women who came to hospital to examination must the same people every 5 years. The same repeat. So the total coverage is in the same group, not expand to the other women who live in the rural area or even in the urban, but they not they don't like to go to the hospital.
Ajahn, it is one question from the chat box. This is anonymous. I don't know who asked the question. He said, "What's about the HPV vaccination in male population?" You have any comment on that?
HPV vaccination in male is the recommended by RTOG in the other groups. So if they have their own budget, they can get the vaccine. But I know that in the other country, for example, in USA, for boys, HPV vaccine are in EPI program.
So could you say a little bit about catch-up vaccination? You normally vaccinate grade 5, but can you tell me what your catch-up program has been, please?
Yes, we start HPV vaccine. The first dose is the 5th grade school girls, and the second dose 1 year apart. So from the 5th grade to the 6th grade school girl in the same school. And because of the COVID outbreak in the past 2 years, the 5th and 6th grade of our country will not have vaccine. So they will have the catch-up program later.
All right. I think we can save the other question for the discussion session at the end. And now, I think, thank you so much, Ajahn Pathama.
Thank you very much.
Now I am honored to introduce our keynote speaker, Professor Douglas Lowy, Prince Mahidol Award, Laureate 2022. Dr. Lowy is the Deputy Director at National Cancer Institute, National Institute of Health in Bethesda, US. Together with Dr. Schiller, Dr. Lowy makes a many groundbreaking contribution to understand the viral pathogenesis of malignancy, especially human papilloma virus. And his work led to development of highly effective HPV vaccine that is clinically used worldwide. And of course, his work has significant implication in preventing cervical cancer, a leading cause of death, especially in developing countries. The topic that he will kindly share with us today is current and future impact of HPV vaccination. It is such a great honor to have him with us today. May I invite Professor Lowy for giving a talk, and please join me to welcome Professor Lowy.
Thank you. Good morning, everyone here at the university, as well as the people online. John Schiller and I are really honored and pleased to be able to talk with you today. And if anybody would like our slides, please just ask for them, and you may have them. I am going to talk about the vaccine, and John will talk about its mechanism of action. But before I get started, I just, as my disclosure, NIH has intellectual property on the vaccine and has licensed the vaccination technology to GlaxoSmithKline and Merck. And John and I are both inventors. I will discuss a potential off-label use of the FDA-approved vaccines, which is fewer doses. And John and I have been involved in other technologies that are not directly related to today's presentation. I'm first going to give you a short discussion of the epidemiology of HPV-associated cancer. The previous introduction really was very good, and I just am going to highlight a couple of things. And mainly, I'll talk about the HPV vaccine, and the take-home messages are it's highly immunogenic, it can prevent mild and serious infection and disease, and it can confer long-term protection even after a single dose. John will discuss the mechanistic basis for long-term high efficacy of the vaccines. And if it is all right with you, we would take questions after both talks. Although if you have a burning question after mine, please feel free to ask it. And during the question period, please feel free to ask questions about something that we might not have discussed, because John and I would like to make this meeting as useful for you as possible. So in terms of the epidemiology, this slide, which is from the International Agency for Research on Cancer, kind of divides cervical cancer into at least 3 different groups. The blue, if you look on the left, the blue represents the incidence of cervical cancer, and the red represents mortality from cervical cancer. And what I've done is to group Africa on the top with the red rectangle, in the middle, South Central Asia with the green rectangle, and then North America down on the bottom with the blue rectangle. And what I hope you can appreciate is that the incidence and mortality rate in Africa of cervical cancer is many times higher than in North America, but still also many times higher than in South Central Asia. And in low and middle income countries, about 90% of cervical cancer cases and deaths occur in low and middle income countries, and cervical cancer represents 90% of HPV associated cancer. If we just look at the United States, we have some disparity. When you look at incidence on the left, the black women are in the red line, Hispanic women are in the blue line, and white women are in the green line. But note that between 2000 and 2019, the incidence of black women and Hispanic women started out being almost twice as high as among white women. And there has been a considerable decrease, but there still is a higher incidence of cervical cancer in black and Hispanic women in the United States compared to white women. If you look on the right side, this gives you different racial and ethnic groups in the United States. The white women have a mortality rate of about 2 per 100,000. And Asian and Pacific Islanders in the United States, if anything, are lower. So this is substantially lower than in Thailand, for example. And black and Hispanic women are higher in their mortality rates than white women. So you have disparities in the United States, but the disparities outside the United States are even bigger. This slide, really you had a wonderful introduction about the different kinds of HPV-associated cancer, and I've emphasized them here, where you have, in women, cervix, vagina, vulva, anus, and oropharynx, and penis, anus, and oropharynx in males. And I think what you should try to take away, I'm going to show this slide again in a few minutes, from this depiction, that cervix is by far the most for women, and oropharynx by far the most for men. And so those are the two cancer types that we really need to be most concerned about for women and men. And as mentioned, we have two different prevention approaches. One is vaccination, which is primary prevention, and we'll talk more about that. And the second is screening, cervical cancer screening, which is secondary prevention. We're not going to talk much about screening, but I do have one slide. Before I get there, I just want to make sure everyone understands that the natural history of HPV infection leading to cervical cancer is the same in high resource settings and in low resource settings. This is a modeling exercise by Emily Berger and her colleagues from the Harvard School of Public Health for the United States. The WHO has said that 4 cases per 100,000 for incidence is what they call the elimination threshold. And you already saw a little bit of that in the earlier presentation. But what Dr. Berger did was to ask, what happens if we increase cervical cancer screening to 90% and keep the HPV vaccination in the United States where it has been? How quickly would we get to the elimination threshold of 4 cases? And then on the other hand, what happens if we keep screening the way it is, but we increase vaccination to 90%? And what the answer is that you get to elimination much faster in the United States by cervical cancer screening than you do by HPV vaccination. I am not trying to say we don't need HPV vaccination. I'm just saying that if you want to decrease the incidence quickly, you should do it with cervical cancer screening. The way John and I often talk about it is that screening is for the current generation of women. Vaccination is for the next generation of women. We'll now turn to the HPV vaccines, and first to tell you about their composition, which John and I were involved in developing this technology. And they are virus-like particles. These are for HPV 16, and we'll come back to the different HPV types in the slide that I showed you previously, but showing it again. And there's spontaneous assembly of 360 copies of L1, which is the major structural protein of the virus, into a virus-like particle. They induce high titers of virion neutralizing antibodies, and they are non-infectious and non-oncogenic. Reinhard Kernbauer, who is a physician at the University of Vienna in Austria, was the postdoctoral fellow who did this research in our laboratory now 30 years ago. This was really the breakthrough finding. If we fast forward to what has been approved by the FDA, there are two first-generation vaccines, Cervarix made by GlaxoSmithKline and Gardasil made by Merck, and one second-generation vaccine, Gardasil 9, that is made by Merck. And as you heard, in Thailand, 16 and 18 account for about 67% or 2/3 of the cervical cancer in Thailand. And that is what's targeted in the first generation vaccine, both with the GSK vaccine and the Merck vaccine. The Merck vaccine is a quadrivalent vaccine because it also targets HPV 6 and 11. Together, they account for about 90%, 90, of genital warts. The second generation vaccine includes these four from the quadrivalent and then adds the next 5 most oncogenic types in terms of their worldwide association with cervical cancer. So this is often referred to as a 9-valent or nonavalent vaccine. And this is the slide that I showed you previously, but here I want to focus on the HPV types rather than the different kinds of cancer. In the dark blue is HPV 16 and 18, and if we just look at cervix, you can appreciate that that's the majority of cervical cancer. The light blue is the proportion of cervical cancer, this is in the United States, associated with those additional 5 types. And in the white, the HPV types that are not covered by Gardasil 9. So about 90% of cervical cancer in the United States and worldwide are directly targeted by Gardasil 9, and Gardasil and Cervarix really directly target about 70%. And what you see down here is in the United States, because we have had cervical cancer screening for a longer time period than you have had in Thailand, our incidence of cervical cancer is about 1/2 of your current incidence of cervical cancer. And so now we actually have more cases of HPV-associated oropharynx cancer in the United States than we have cervix cancer in the United States. And about 3/4 of the oropharynx cancer in the United States occurs in men, and about 1/4 in women. But if you just take all the total here for men and the total here for women, in the United States, about 60% of HPV cancer is in women, whereas about 40% are in men. In Thailand and many other countries, it's a much higher percentage of HPV-associated cancer in women compared to men, mainly because number one, the incidence of cervical cancer is higher, and number two, the incidence of oropharynx cancer is lower. So what are the principles, the goals of HPV vaccination? And we think of there being 2 goals. The first is to directly reduce the risk of infection and disease in vaccinees But the second is indirectly to reduce the risk by reducing the prevalence of the HPV vaccine types in the general population. This is often referred to as herd immunity or herd protection. And for any preventive vaccine, you're going into a normal population. Only a minority of people actually are going to benefit from the vaccine because only a minority of people are destined to get the cancer. And so safety becomes of paramount importance. So these are two published papers from the United States looking at the safety profile by the Centers for Disease Control, the United States, CDC, and others. First, for Gardasil, basically no confirmed safety signals identified in 2016, and last year, following the introduction of Gardasil 9, no longer-term findings support the existing robust literature on vaccine safety for the 9-valent vaccine. What about some of the characteristics of the vaccine? And if we first say the vaccine has very high efficacy, more than 95% against the targeted HPV types, and long duration of protection, more than 10 years of protection. Second, the vaccine confers sterilizing immunity. What this means is that it prevents infection in most vaccinees. The vaccine induces herd immunity even with suboptimal vaccine uptake. And I'll show you data from the United States because unfortunately, we have not been a high uptake country up until quite recently, but there's very good evidence for herd protection among women, as I will show you. And it's very important to realize that while the vaccine works very well for preventing infection, as far as we can tell, it does not alter the natural history for people who have prevalent infection. So the vaccine needs to be given before people become sexually active because it's primarily a sexually transmitted infection. And women, at least in the United States and other high-income countries, tend to become HPV positive very soon after becoming sexually active. So this is the slide that talks about herd immunity in the United States. And this is for 14 to 24-year-old women, 12 years after HPV vaccine introduction in the United States. So basically, it's looking from 2007 to 2018 or 19. And what to focus on first is the blue bars, where on the left shows at the beginning of the introduction of the vaccine, 2007 to 10, then 2011 to 14, and 2015 to 18. And the blue bars are the unvaccinated women, not the vaccinated women. These light blue bars are the vaccinated women. And so if you're looking, is there herd immunity, you can see a progressive decrease in the prevalence of the vaccine types. This is the 4 types in Gardasil. And how does that correlate with the uptake of the vaccine? So the thin dotted line that starts 2007 to 10, if you look also on the right for the vertical axis, this is the percentage of women who got at least 1 dose. We are not as good as you are at getting the subsequent doses. So you have a fall off of about 15% or so between the first dose and the subsequent doses. We have a fall off of about 30% or 35%. So this is just looking at 1 or more doses. And in 2007 to 10, it was about 30% of women, and by 2018 went to 60% of women. So that's just with one dose. And at that time, you have substantial herd immunity. In the United States, we started vaccinating boys in a real way after 2011, because that was when the FDA approved the cancer prevention indication for boys. And you'll note that in 2011, it was 15% of boys and 30% of boys in 2018. So there's so you can look forward to developing herd immunity in Thailand because your uptake is substantially higher than in the United States. What about cervical cancer? The interval between infection and the development of cervical cancer is usually at least 15 years or longer, but in the last few years there have been data from several countries, including Sweden, Denmark, and England, using either Gardasil or Cervarix, the Merck vaccine or the GlaxoSmithKline vaccine. And these are data from Denmark, just showing that if women have been vaccinated when they were 16 or younger, there is about a 90% decrease in this observational countrywide study for the development of cervical cancer. The red line are the women who have been vaccinated, and the black line are the women who have been unvaccinated. If you turn your attention all the way to the right, these are the women who were vaccinated when they were 20 or older, and there is no substantial benefit. In the long run, we will see some benefit, but this emphasizes how important it is to vaccinate women before they become sexually active. And the same is true for males in countries where males are being vaccinated. And if you look in the middle, it basically is in between. There is a little bit of protection, but nowhere near as much as the women who were vaccinated in general before they initiated sexual activity. SARS-CoV-2 infection has, of course, been a terrible scourge for the world over the last few years. And I think it's instructive to compare the HPV vaccine in some respects to the SARS-CoV-2 vaccine. And I'm talking about the messenger RNA vaccines for SARS-CoV-2. The HPV vaccination prevents even benign infection in most vaccinees. SARS-CoV-2, many vaccinees remain susceptible to mild infection. We are all, I assume that almost everyone here in this room has been vaccinated, but you are wearing masks because of the risk of getting, hopefully, mild infection rather than serious infection. Vaccination readily induces herd protection with HPV vaccination, whereas vaccination has not been shown to induce herd protection for SARS-CoV-2. The HPV vaccine confers strong protection against all variants within a given HPV type. That's very important because worldwide there's considerable variation within a type, but the evidence is quite strong that it doesn't matter which variant you have for HPV vaccination. By contrast, we are all painfully aware that the SARS-CoV-2 vaccine may confer reduced protection against some variants. The vaccine for HPV induces more than 10 years of strong protection, whereas two doses provide several months of protection for SARS-CoV-2. The last topic I want to discuss is, might a single HPV vaccine dose confer years of protection? And as you heard, Thailand is already doing important research, really in a real-world setting, using clustered trial design to look at 2 doses versus 1 dose. So why might one want to have just 1 dose? And the issue really is logistics, cost, and what happens if you don't do it? The HPV vaccine was approved more than 15 years ago, 1-5. But in low and middle income countries, only about 10% of eligible young women have received the vaccine. And it's been demonstrated in many countries with low resource settings, there is a big fall off if you need to give the second or third dose. So, and our proposed solution is single-dose vaccination. It's less expensive and logistically easier than 2 doses, but not yet standard of care by the FDA, but it recently, as I'll show you in my second to last slide, has become standard of care as an option for the World Health Organization. We published a couple of years ago that 1 vaccine dose in our Costa Rica vaccine trial could prevent HPV-16 infection as effectively as 2 or 3 doses 11 years after vaccination. This is with the GlaxoSmithKline vaccine, and with the IARC India trial of Gardasil, there are analogous data published in the following year. 100% of the women who received 1 vaccine dose remain positive for HPV-16 and 18 antibodies, and John will show you the data from the paper. So there currently is a good deal of research going on in many parts of the world, including Thailand. I just will emphasize that we have this post hoc analysis of our first Costa Rica vaccine trial. We have the second trial that we call Escudo, which is comparing 1 dose and 2 doses of Cervarix or Gardasil 9. And Amy Kramer and John from the NCI have had major input into the Kenshi trial, whose results were reported last year for 18 months and had more than 95% efficacy. And this led the Strategic Advisory Group of Experts on Immunization, known as the SAGE Group, to make a recommendation of 1 or 2 doses for girls aged 9 to 14. Previously, previously it was only 2 doses, 1 or 2 doses for young women, 15 to 20. It was previously 3 doses. And then these other recommendations for women above 21 and for immunocompromised individuals. So my last slide is really to try to summarize. First, basic research led to identification of HPV as the cause of several cancers and to development of HPV vaccines. Second, virus-like particle display is highly immunogenic. It can induce herd immunity, durable protection, and prevent cervical cancer. Third, if the HPV vaccine is FDA approved for a single dose, it will be the first subunit vaccine to achieve single dose approval by the FDA. Most important, a global single-dose recommendation, we believe, could make it feasible to control the worldwide public health problem of HPV-associated cancer. Thank you so much for the invitation, and looking forward to hearing from John.
Thank you very much, Professor Lowy. It's a very magnificent talk. And as Professor Lowy has requested, please hold your questions until the end of Dr. Schiller's talk. Okay, so thank you very much. Now it's also my privilege and honor to introduce Professor John Schiller, Prince Mahidol Award 2022 laureate. Dr. Schiller is currently a NIH Distinguished Investigator and Section Chief in the Laboratory of Cellular Oncology, Center for Cancer Research, National Cancer Institute, NIH, United States of America, or USA. Dr. Schiller has studied various aspects of papillomavirus molecular biology, immunology, and epidemiology. The laboratory headed by Dr. Schiller and together with Dr. Lowy has led in the discovery, characterization, and clinical testing of virus-like particle vaccines to prevent the HPV infections that cause cervical cancer and other cancers. The topic that Dr. Schiller will share with us today is why the HPV VLP vaccines are so effective. Without further ado, may I invite Professor Schiller to give a talk, and please welcome Professor Schiller.
Well, it's indeed a pleasure to be here to talk to you today, and particularly it's a pleasure to hear how well Thailand is doing in their cervical cancer control. It's really, I think, a model for many countries in terms of how to tackle this problem. But what I'd like to tell you today is about why the vaccines work so well. And it's really been remarkable, and it's been a privilege of a lifetime to be able to be involved with this vaccine, because it has basically exceeded all reasonable expectations for a vaccine right from the beginning. And so it becomes an extremely interesting question is, why is that? Why does this vaccine work so well? And so I'd like to go through today and tell you that it's, as most things in biology, it's multifactorial. But we think that the three main reasons why it works so well are shown here. First of all, we're very fortunate that HPV is has a DNA containing genome, which doesn't evolve rapidly as RNA genomes, for instance, in HIV or hepatitis C or the SARS, as we're all seeing. And so that it doesn't escape antibody-mediated protection rapidly like those vaccines. Secondly, the vaccine is really exceptionally good at inducing neutralizing antibody responses. And I'm going to show you in a minute why we think that that's the case. And lastly, and again, we didn't know this when we developed the vaccine, is that the way that this virus infects makes it just uniquely susceptible to neutralizing antibodies. And I'll go over that as well. So really, one of the important things is that I think that all this sort of basic science provides plausibility as to why the HPV vaccines are the first subunit vaccine to induce long-term protection after a single dose. And I think that's very important as we think about, because there's a lot of skepticism that a one-dose vaccine can work. And biological plausibility, I think, is important for this. So first of all, in terms of immune response, it has a remarkable consistency of seroconversion. And this is a recent trial where the girls got Cervarix or Gardasil 9, and what you can see is that for all the different types, regardless if you get 1, 2, or 3 doses, there's essentially 100% seroconversion. There's essentially no non-converters. And even more remarkable is if you look at the decay of the antibody responses. Most vaccines, they continue to drop, like for instance the SARS vaccine. But here what you can see that even after a single dose, between year 2 and year 11, there's no change in antibody titers. They're completely flat. And this isn't just true for geometric mean titers, but actually for individuals as well. And we actually have unpublished data now out to 16 years. And again, there's no statistical change in titers between year 2 and year 16. So this really explains why we have such long-term protection for this vaccine, because the titers of antibodies don't drop. And one of the questions we had when the data that I just showed you was for Cervarix, which is what we studied in this Costa Rican trial. And one thing that Doug didn't mention is that Cervarix has a special adjuvant. It has a TLR agonist. It's monophospholipid A, which is a detoxified form of LPS. And so we were concerned that this ability to have this plateau level was unique to having that adjuvant. But recently it was published that if you compare Gardasil 9, which has a simple alum adjuvant, with Cervarix, which has this complex adjuvant, although the titers for Cervarix are somewhat higher, if you look for a single dose, it also plateaus for the simple adjuvant. So you don't need a complex adjuvant in order to get this to happen. So the question is, why does it happen? Now, one of the questions that comes up is that, well, if you get this constant level of antibody titers, is it because the women are getting exposed to the virus from their partners or other types of environmental exposure, and that's what's keeping the antibody levels stable? And it's almost uncertain that that's not the case, because if that would happen, what you'd think there'd be a bimodal distribution of antibody titers, where some of the women would be losing the titers, whereas other ones, because they're getting environmental exposure, would get boosting. So you'd have a bimodal distribution. We actually looked at this for individual titers, and it's the most nice distribution, as you can imagine, that's centered around 0. So there's a few outliers you can see here that are more than 2 standard deviations away from the mean. So we think that there can be environmental boosting, but it's very rare, okay? And that the majority of the women are maintaining their antibody titers not because they're being exposed to antigen, because they have long-lived plasma cells that are continuing to pump out antibodies for the rest of their lives. So why are they so immunogenic? And we think that the 3 important things are structure, structure, and structure. And it's quite unique and ironic that we developed the virus-like particles because we thought we needed to have conformationally dependent epitopes. So we had something that looked like a virus, it would have the right shape of the epitopes on the surface, whereas denatured proteins didn't work. We knew that. But Martin Bachmann and Ralph Zinkernagel came out with this hypothesis just a year after we started this work with the virus-like particles, and said that they believe that B cells specifically recognize particular antigens with an epitope spacing of 50 to 100 angstroms as foreign. Now, why did they postulate this? The reason was because this epitope spacing is commonly found on microbial surfaces, like the surface determinants of a virus like papillomaviruses, and also on other microbial structures like bacterial papillae. And so protein complexes of this spacing rarely occur in vertebrate animals on our own proteins, on our own body surfaces. And so I like to think of the B cell receptors as having evolved as an antigen-specific pattern recognition receptor. And so mechanistically, what we think that happens is that if B cells, like naive B cells, interact with soluble protein, which is kind of the form of protein that's floating around in our bloodstream all the time, they don't react very well to it. And so you get relatively weak activation signals and low levels of antibodies. But when these B cell receptors, which are just the antibodies on the surface of the B cells, interact with this polyvalent oligomerization, you get downstream signaling because you get oligomerization of the associated tyrosine kinases and get a strong survival signal and proliferation, high levels of antibodies of long duration. So really this repetitive antigen structure is really the decision to invest in long-lived antibody responses because it's a signal for foreignness. And so now one of the things we're trying to understand is what really is the difference in signaling when a naive B cell sees these polyvalent repetitive structures versus a simple structure. And hopefully if I come back in a couple of years, I'll be able to tell you the answer to that. But there are some other advantages that VLPs have in addition to this increased activation of B cell receptors. They're just the right size particles to be efficiently trafficked to the lymph nodes. If you start having bigger particles like a herpes virus virion, they can't go directly to the lymph nodes. They have to be taken in antigen-presenting cells. But these 55 nanometer particles are just perfect for getting there by themselves. And because, again, this polyvalent nature, they're readily phagocytized, and so they induce strong T helper cells. And another aspect is that their polyvalency leads to stable binding of natural low-emitting IgM and complement, which promotes their acquisition on follicular dendritic cells. So they're presented to the naive B cells in a very immunogenic way. So what are the lessons for the COVID vaccine? I think Doug and I were both doing comparisons to the COVID vaccines because everybody's interested in. Well, I really think that there's no better way of inducing long-lived antibody responses and high-potency antibody responses than virus-like display. And so that virus-like display of the RBG binding domain is likely to be the way to most consistently induce high levels of antibodies and long-lasting antibodies. And so we're very gratified that some companies and some academic groups have taken up the gauntlet and tried to develop virus-like display vaccines for COVID. And one vaccine that I think is very, very promising is this capsid-like display of the CoV-2 receptor binding domain that's linked to a phage particle, okay? And you can read it here. It induces much higher levels of antibodies than the typical RNA vaccines or the adenovirus vaccines. So I think the future of the SARS vaccine is actually a virus-like display vaccine. Now, one thing that it's important to know is that not all virus-like particles are created equally. So part of the reason that HPV responses were so unexpected is because previously people have been studying hepatitis B, which is also a virus-like particle. It's smaller, it's 22 nanometers, and it doesn't have the same immunogenic characteristics, whereas one dose doesn't induce seroconversion quite often, and even 3 doses, the titers continue to wane. Now, it induces a good memory B cell responses, and so that it doesn't necessarily protect from initial infection, but it protects from disease because you get an anamnestic response. And the same is actually true for a more recent hepatitis E vaccine, which is also a 20 nanometer virus-like particle. And so one of the questions for the future is what is structurally wrong with these virus-like particles? Is it that they're not rigid lipoprotein structures? Are there too few repeats? Is it too small? It's important to find this out to develop virus-like particle vaccines for the future. And one of the things, it may not just be tightly spaced epitopes. And one thing about the HPV virus-like particles is they're really locked down. The platform is very rigid, and the determinants that induce neutralizing antibodies we call loops, but they're actually, again, very locked down solid structure. They're very resistant to denaturation. And so the amount of rigidity that's needed in the platform and also the antigen is still not completely worked out. We think that these are important elements. So one of the big things that came up when we said, well, we think one dose vaccine works was, well, no, the quality of the antibody must be worse because everybody knows you need to have boosting in order to generate high quality antibody responses. And that's absolutely not true, okay? And the VLPs prove that. And so if you look at the quality of the antibody responses by looking at the ratio of binding antibodies, which may be neutralizing, they may be high avidity, maybe low avidity, to neutralization, which is really the functional antibodies, you can see there's a 1-to-1 correspondence for 1 dose and 3 doses. There's no difference in the ratio of the two. So there's no difference in the quality of the antibody responses. The magnitude is lower, about 3-fold or 4-fold lower, but they still are very, very good responses. And the other way is to look at avidity. Avidity is just the summation of the binding strength of a polyclonal response. And one of the ways you can look at this is what's called a chaotropic ELISA. So you do a regular ELISA, but now you use guanidine hydrochloride or something else that loosens up the interactions between molecules, and you see what dose you need or concentration to get 50% to fall off. And again, what you see here is that if you look at 3 doses, 2 doses, or 1 dose, whether it's Cervarix or Gardasil 9, there's no difference in avidity for either HPV 16 or HPV 18. And so again, this really indicates that the quality of the antibody responses doesn't go up with boosting. And so this raises the question is during boosting, where are the long-lived plasma cells coming from? So long-lived plasma cells are the types of B cells that after activation go to your bone marrow and pump out antibodies for the rest of your life. And so now what we've been able to show is that after prime, with the VLPs, we know we get long-lived plasma cells because now out to 16 years, the titers don't change. We also know that they induce memory B cell responses, both short-lived and long-lived memory B cell responses. So now the question is, now when you boost, where do the long-lived plasma cells come from? Do they come, as most people would have thought, from the memory B cell pool re-entering the germinal-Lacena reaction and generating long-lived plasma cells? Or do they come by the same pathway through naive B cells that occurs when we do the prime? And we think that the latter is probably happening because, again, the quality is not changing, okay? We're basically doing the same thing over and over again. And this is something that most people, B cell immunologists, would not have thought to be occurring. One interesting thing we found is that we asked the question, well, what happens if a woman already has HPV antibodies from a natural infection? How does that affect her response to the vaccine? And what we found is that it doesn't affect the titer, the amount of antibodies she ends up getting, but it affects relatively largely the avidity of her antibody responses. So first of all, if you look at the avidity after natural infection, which again is being, the antigens being exposed on a mucosal surface, maybe not even going through a germinal center reaction, they have very low avidity. So this is the dotted line right here. But then after vaccination, if you haven't been exposed, you have very high avidity coming up and then being maintained out to 11 years. If you were previously vaccinated, previously had an infection and have onboard antibodies, you can see your avidity increases substantially, but it never reaches the level, okay? Now, we don't think this is a problem for HPV because it doesn't matter whether you have onboard antibodies or not, you're protected long term. But we wonder if this could be a problem for vaccines like SARS, where if you have an inapparent mucosal infection like HPV, and now it's a SARS vaccine in your respiratory tract, is the quality of your antibody responses to the vaccine going to be as good? And I think this is something we really don't know yet, but it's something that we should probably start to look at. So now we talked about immunology, but now let's talk about the second thing I mentioned, which is virology and the mechanism in which the virus infects. And again, we didn't know anything about this when we developed the vaccine, but subsequently we were looking in a mouse vaginal challenge model using HPV vectors, which instead of real virus, but like HPV-16 vectors, we looked at the mechanism of infection and how antibodies prevent that infection of a tissue, cervical vaginal tissue. And what we found is surprisingly that the vaccine, the virus doesn't bind to intact epithelium, cervical vaginal epithelium, which was very surprising because this is a tissue it infects. And it's not just in mice, but we did this in non-human primates, and it was exactly the same. So initially, the virus has to bind, it can only bind to the basement membrane at sites of trauma or permeabilization. And what they bind is specifically modified forms of heparin sulfate proteoglycans, and these modifications are specific for the basement membrane. They don't occur, even though the surfaces have HSPGs, they have different modifications, so they don't bind. And then upon binding, this induces a conformational change in the virion. They kind of open up and expose part of the minor capsid protein, L2, to go pop out and get clipped off. And then that exposes a surface on this major capsid protein, L1, that can then, and only then, bind the keratinocytes and initiate infection. And from the point of view of neutralization of antibodies, is this whole process of sitting on the basement membrane and even attaching to cells takes hours. So what you can imagine is that now there's a lot of time for the antibodies to do their thing, to work. It's by far the slowest virus in terms of infection that we've ever seen. Most viruses, once they get on their cell surface, it's within seconds or minutes they get internalized. And so this just shows you some of the data. So this is one of these, what we call pseudoviruses, where instead of the real virus, it's got a reporter gene, in this case, red fluorescent protein. And then the trick is we label the outside with Alexa4 488. So the particles themselves are green. And if we get infection, then those cells turn red. And these particles are fully infectious. And this just shows that in the vaginal tract, whether it's squamous epithelium or simple columnar epithelia, the type of epithelia they infect, there's absolutely no association with the cell surface. But this is one of the most amazing pictures is when you see as soon as you traumatize the epithelium, so here's the basement membrane, there's no binding to the apical surface or even the basolateral surface of the epithelium. The only thing they bind is this basement membrane. Just very dramatic. And then after a period of about 24 hours, you see transfer, it's a little bit hard to see in the light, but you see transfer from the basement membrane to cell surfaces, and then within about 2 days, you see red cells, which are the infected cells. So it's, again, a very slow process. And so what this allows us is that it gives another mechanism for which the antibodies can effectively reach the sites of infection and prevent infection. So this vaccine is delivered intramuscularly, and as most of you know, that's good for generating systemic IgG, but really bad for generating local mucosal IgA. But how we thought this vaccine was going to work is that the female reproductive tract is fairly unusual and that there's quite a what's called transudation of systemic IgG into the cervical vaginal mucus. And this happens by a receptor. It's the neonatal FC receptor, which is the same receptor that pumps antibodies from the mother across the placenta into the infant or into the fetus. And so we thought it was going to work by that way, but we were a little concerned because if you measure the levels of IgG, VLP specific in women after vaccination, they're somewhere between 10 and 100-fold lower than serum concentrations. But now that we know that you need trauma and exposure to the basement membrane to get infection, what that means is that the virus is trying to get down to this basement membrane against a gradient of exudated capillary and interstitial antibodies that may actually be very close to the level that's in the blood. And we know that this is a sufficient mechanism for protection because the vaccine is very good at protecting against genital warts. And many genital warts occur on surfaces that aren't bathed by mucus. You know, they're dried cornified skin. And so that we think that this is actually the main mechanism, is this direct exudation of interstitial antibodies that leading to protection. And so what are the lessons for the COVID-19 vaccine? And again, this is going to be problematic because inducing sterilizing immunity is going to be much more difficult. Doug mentioned that this vaccine surprisingly induces sterilizing immunity. You don't get an infection at all. COVID directly infects apical surface of respiratory epithelium, so there's not going to be antibody exudation at the site of infection like we see for HPV. And there's also not extensive transudation of systemic IgG in the upper respiratory tract, although there is in the lower respiratory tract. And that's probably one of the reasons why it protects against fatal infections of the lungs better than it does to protect against upper respiratory tract infections. So one of the questions we asked for the HPV, maybe I failed to mention, but so one dose, the concentration of antibodies is about 4-fold lower than it is after 3 doses. And so we asked the question, well, is the level of antibodies that are induced by vaccination just on the edge where it's barely protecting, or are we way over the minimal level needed? We couldn't do this in women, but so we did do this in mice. And what we did is sort of a classic experiment, which is called antibody passive transfer experiment. So we vaccinated a rabbit, in this case 3 times, and we took out the serum from that rabbit and then diluted it and put this diluted sera into a naive mouse. So the mouse hadn't been vaccinated. And then we challenged that mouse intravaginally, again with a pseudovirus, in this case expressing luciferase. So if you get infection, the light is emitted when you put this luciferin. And if you get protection, then you don't get any light. And what we're able to show, and this is the most amazing result I think I've ever had in biology, is that we're able to dilute the sera from that rabbit. So it'd be the circulating sera in this mouse is a 10,000-fold lower than what the circulating concentration was in the rabbit, okay? So you would probably estimate then that probably a 4-fold difference isn't going to make a whole lot of difference. Now I have to admit that rabbits induced about a 10-fold higher level of antibodies than either mice or humans. So if you believe this analogy, then humans only have about 1,000 times higher level of antibodies on average than needed for protection. So that's one of the reasons why we think that we're seeing such good protection with even a single dose, because the levels are so high in comparison to the levels needed to protect against this particular infection. And just to illustrate that, this shows that if you look in the same mice and you say, what level of antibodies do you need to protect from infection with these pseudoviruses in a dish? So in vitro neutralization, which is the standard thing people look at to determine protection, versus what's the level of antibodies you need in the animal to protect against high-dose challenge, you can get protection in vivo with levels of antibodies that are about 100-fold lower than anything you can measure in an in vitro assay. So what does that suggest or indicate? That there are mechanisms of protection that occur in vivo that aren't being monitored in the in vitro assay. And we actually then looked, and this is one of the few times this has been done in the animal to just show what's the mechanism of neutralization in the tissues that are normally being infected by the virus. So again, turning to the cervical vaginal challenge model, I'll just show you, summarized in cartoon, the data, is that if you use high levels of antibodies, only like 100-fold dilution, then you don't see any association of the virus with the basement membrane. So it's very much what you see in vitro assay, where you have enough antibodies, it just coats the surface of the virus, and it can't interact with its primary receptor. But what was interesting is at low levels of antibodies, we actually didn't disrupt this initial interaction with the basement membrane on these heparan sulfate proteoglycans. The virus was even able to open up and expose these minor capsid protein epitopes. But what we never saw was stable association with the cells, okay, the keratinocytes that were moving in. But where we saw that all the virus was collecting was on neutrophils and monocytes, okay? So what we postulate is happening here is that these, oops, I don't know why that went up. Oh well. So what we postulate was happening there was that the antibodies serve as handlebars, and the neutrophils and monocytes have Fc receptors. And so while these viruses are sitting on the surface, the antibodies can serve as a link between the phagocytic cells and the virus, and they can just pull them off before they have time to go and infect the cells. So in conclusion, the HPV vaccines are very effective at preventing incident infection and disease by the vaccine types. And the reason that they are is because the VLPs are exceptionally potent inducers of neutralizing antibodies. And as I just mentioned, the viruses are exceptionally susceptible to inhibition by antibodies. We believe that these observations provide important biological explanations as to why the HPV VLPs may become the first widely employed single-dose subunit vaccine. In addition, they provide a roadmap for developing vaccines against other infections that could similarly induce potent and long-lasting antibody responses. So I'd just like to end with acknowledging a wide number of collaborators who have been involved in this work. And of course, Doug Lowy at the top of the list. We've been working together on this for the last 20 or 30 years, but also past members of the lab and also many very talented investigators, both in terms of clinic and in the laboratory. So with that, I think I'll stop and maybe have Doug come up here and we can take some questions.
Thank you very much. Any question from the floor? Yes, please, อาจารย์กุลกัญญาค่ะ
Thank you. Thank you, Professor Laureate and Professor Schiller. Very excellent lecture.
Okay.
Yeah.
Sorry, I couldn't see.
I'm Kun Kanya. I'm a pediatric infectious diseases. So from your elaboration, can we just ignore the level of antibody induced by the vaccine? As you can imagine that the manufacturers claim the superiority because of the higher immunogenicity. As well as the single dose, although is quite equivalent in terms of protection, and that cannot be proven by statistical significance. However, there's a trend of a little bit lower in overall efficacy among the single dose trial or the single dose group of the experiment. So even as of now, WHO didn't recommend single dose as the primary, but recommend as an alternative regimen. So can we, and if we collaborate that with the level of antibody that, has shown that the single dose has lower, although perhaps good enough antibody levels, could that, should we ignore that, or can we just say it's okay for the level of antibody, again, from single dose or from different manufacturers.
Do you want to take it? Because I've been talking a lot.
Yeah, thanks very much for this question. First, I think that efficacy is what is of paramount importance, and the Costa Rica vaccine trial that we're currently doing will be looking after 4 years, which is how all of the HPV vaccine approvals have been given previously. And if there is high efficacy for one dose, similar to 2 doses, then I think that is number one. Number two, we don't know what the minimum antibody level is for required for high protection. But if there is as high protection as, for example, seen with the Kenshi trial, and that goes on for at least 4 years, at that point, I think that we will be able to say the minimum needed or is not, whatever is 1 dose for the Merck vaccine, because as John mentioned, it's less immunogenic than the GSK vaccine. That is, if you will, a sufficient dose of antibody. I think that another question which arises, though, is what about biosimilar vaccines? And then, to me, immunological equivalence is going to be important. So if you did immunological non-inferiority trials comparing, let's say, a biosimilar vaccine to Merck or a GSK vaccine, depending on the adjuvant, you could, I think, extrapolate from those results. This is, of course, our speculation, but we think that it is reasonable. John, did you want to comment?
Yeah. So once we establish that 1 single dose vaccine in a randomized controlled trial works, then that can be used as the standard to do non-equivalency, immune non-inferiority for other biosimilar vaccines. And the other thing I would add is right now with our assays in vitro, we think that the level needed is below the level that we can measure in our in vitro assays, but we're actively trying to develop in vitro assays that more mimic what's happening in vivo in terms of having phagocytic cells pull it off the basement membrane. And so we're actually trying to get more assays that can have higher sensitivity for those lower levels of antibodies.
Yes, please.
Okay, I'm Dr. Mongkon from gynecologic oncologist at the Siras Hospital. I have 2 questions, but I do it one at a time. The first question is, at the beginning of the global introductions of the HPV vaccinations, we heard about the cross-protections of the vaccine, and they try to compare of the cross-protection between the bivalent cervix and the Gardasil. And nowadays, I think we don't hear about that. What is your comment about the cross-protection?
Yeah, I think that cross-protection is substantially greater for the GSK vaccine than for at least Gardasil vaccine. There hasn't been as much evaluation of Gardasil 9. But Gardasil 9, because it includes the HPV types where the GSK vaccine has been shown to have cross protection. In that sense, if you will, it becomes somewhat less important. It's not that cross protection is unimportant. It's simply that the number one priority is the HPV types that are specifically targeted. Having said that, if you were to have, let's say, a vaccine that was analogous to Gardasil 9 with a stronger adjuvant like the GSK vaccine, you might see more cross protection, so that instead of protecting against, let's say, 90% of potentially oncogenic infections, it might protect against an even higher proportion. But there's no strong evidence yet for or against that theoretical possibility. John?
And it's important to point out that cross protection isn't as complete as type-specific. Type-specific protection is essentially 100%. Cross-protection for types goes from 0 to, at best, like 30 to 50 or 60%. So it's not 100%, even with the GSK.
Okay, thank you, Professor. My second question is regarding the single-dose vaccinations. We have heard, so far as I know, we have the data, the efficacy data from the Cervarix, Gardasil 9, but we don't have the data for the other vaccine. So can we just apply this efficacy data to the new and cheaper upcoming vaccine, like from China, from India?
Yeah.
Can we?
Yeah, so what's probably going to happen is, again, once we establish a level of antibodies for the Glaxo and Merck vaccines that correlate with protection, okay, then what the Chinese and Indian vaccines are going to do is do immunobridging and show that they generate the same level of antibody responses. And some of those studies are already undergoing, okay?
Okay, thank you.
Yes, Dr. Piraipan ค่ะ Yeah.
Thank you. You mentioned about the importance of neutralizing antibodies, but HPV is non-cultivable virus. How do you measure the neutralizing antibody? Most of the study rely on ELISA, that is the binding antibody, not the neutralizing antibody. How can you detect neutralizing antibody? I think this is important if you decide to switch from multi-dose to single-dose vaccine. And also for the
So that's a very reasonable question. We have found that the kappa correlations are very high. They're essentially close to 0.9 between the ELISA and the neutralizing activities. So we're reasonably confident. But it is important to confirm neutralization for a subset of patients when you're doing something like that.
Yeah, any new product, you have to show that there's this direct correlation between neutralization and ELISA. And once you establish that, then you can use an ELISA as a surrogate for neutralization. But you have to do neutralization initially to show that it is correlated. Absolutely.
Dr. Nakorn, please.
Thank you very much, Professor Douglas Louis and Professor John Schiller. I am Nakorn Premsi, Director of National Vaccine Institute of Thailand. And thank you very much for the presentation. I have two questions. Dr. John, you mentioned a little bit on the natural booster, kind of natural infection booster. Can you elaborate a little more on that? Is there any evidence that the people who got vaccination and maybe later infect with the HPV virus, that we can get more a booster effect of that kind of thing?
Yeah, there really isn't any good data, in part because we really think that real infection after vaccination is vanishingly rare. That when we do see what are called breakthrough infections, they almost always occur at the beginning of a clinical trial, in the first 6 months or a year. And what we think they are is emergence of prevalent infection. In other words, they were there at the time we vaccinated, but we just didn't detect them even with PCR. We just missed them. And so if you start going out longer periods of time, you almost see no breakthrough infections. And so that class we don't really see. And so what we guess is those few women who look like they're getting an anamnestic response because their titers are going up in later years are probably, it's due to exposure of virions from their infected partner, okay? But we think it's not very common. Now, the female reproductive tract is actually not a good place for inducing antibodies. And the reason why the female reproductive tract has evolved that way is presumably because anti-sperm antibodies is one of the leading causes of infertility. If there's anything that's been selected for in evolution, it's fertility, okay? And so it's a very poor place to try to generate an antibody response. And that's why after natural infection, the women are shedding these antibodies, but it takes a year, 6 months or a year, for women to seroconvert to their own infections. They have an infection, and then only about half of them ever seroconvert, okay? So that's one of the reasons why we think that natural boosting like that doesn't occur very much.
And in Costa Rica, which is where our data come from, the prevalence of HPV-16, which is what John showed you the results for, the prevalence is relatively high, but that is not true for HPV-18. HPV-18 prevalence is low, and the profiles that you see for 18 are virtually indistinguishable for the profiles that you see for 18. They're very similar, and it's not plausible that women are getting exposed to 18 with any frequency.
I have one more question. Please see if the time allowed. I'm interested in the correlates of protection and the immunobridging study. So would that be established by the FDA that the following after vaccine, maybe the new vaccine can be developed by using the immunobridging non-inferiority study? That what it has been established by the FDA yet or not?
So the FDA has not, they have accepted immunobridging in one aspect. So the trials were all done in 15 to 26-year-olds, okay? But we wanted to vaccinate 9 to 14-year-olds, and no efficacy trials have been done in that age group. But what the company showed is that their antibody responses in neutralization in ELISA assays are as good as in the efficacy population. And so they have accepted immunobridging in those cases. Now, in terms of across vaccine, products, it's my understanding that the Serum Institute product in India is being approved in India based upon immunogenicity bridging to Gardasil, okay? That they're not doing an efficacy trial. So the individual, regulatory agencies could maybe make different decisions based upon that.
10 years ago, John and I and others were involved in organizing an experts meeting with NCI and IARC, the International Agency for Research on Cancer. And and the goal of the meeting was to discuss in a rigorous and collegial manner what the future studies should be for endpoints. And that was published in 2014, and it included immunobridging under certain circumstances. And the FDA has essentially embraced what the recommendations were there.
Let me ask. Okay, Ajahn, please.
I'm Vishai, GYN oncologist from Chulalongkorn University. I enjoyed your talk very much. Thank you so much. I have two questions for you. The first, I think that we have some study showing that because the HPV vaccine is the prophylactic vaccine, but they have some study showing that they have like a therapeutic effect. For example, in genital wart or in some skin disease or something, can you have the explanation for this study?
Yeah, so we actually don't feel that it has therapeutic activity, and we never thought it would, because this is the protein there is L1, okay? And L1 isn't made in the basal layers, the lower layers of the epithelium where the infection is being maintained. It's only being expressed in the terminally differentiated dying keratinocytes, which aren't under much immune surveillance. And so even though it is able to generate some CTLs, the virus-like particle vaccine, it doesn't eliminate infections. And in the clinical trials, it was very clear that it neither caused regression nor stopped progression of primary infections. And that's the reason we think that it doesn't act that way. Now, there has been some cases where it's been reported where you can vaccinate after lesion clearance and you prevent new lesions. But I think that's what you're really doing it's acting prophylactically against a new infection. Okay, it's not actually acting therapeutically.
Okay. Thank you for my second question, because I think that we don't have the exact level of the minimum protective level of the antibody. So I don't know that for the single dose, if we, I'm really glad to hear that the single dose should work, right? But anyway, that depends on the adjuvant of the vaccine, because I'm aware that you show the data is either any vaccine can work for the single dose, but I don't know because we don't know the minimum protective level. Is that the type of the vaccine play some role?
Well, I don't mean to push back, but to me, it's good news that we don't know what the minimum level is. The way that minimum levels are usually established is because of vaccine failure, and you identify differences in the recipients of the vaccine where it failed versus the recipients where the vaccine was successful. And from your perspective, it may be unfortunate that we don't have vaccine failures, but from our perspective and some other people's perspective, it's not such a bad thing that there aren't vaccine failures.
Thank you.
All right. I think I will have one last question for this section from the audience online.
Sure.
I think this probably from a T cell immunologist. He asks are there any role of T cell because you mentioned solely on the antibody.
Yeah.
Please.
So again, that what I mentioned is that, again, we think because L1 isn't expressed in the lower level, that the T cells that are generated, and we do generate T helper responses, and we do generate CTLs to a certain extent, that they don't play a role in clearance, and that's why it was never predicted that they would act therapeutically because of that.
Yeah. I should point out that the passive transfer studies that John talked about in his presentation clearly are just looking at humoral immune function, not at T cell function, and it seems to parallel the protection that is seen with the vaccine.
Yeah, even in terms of type-specific protection, the passive transfer says that the antibodies are sufficient to confer protection.
All right, thank you very much.
Thank you.
We will save a lot of questions for the discussion part later, but it's time for a break.
Yeah.
So the organizer provide a complimentary break outside the room, and please come back around 10:45.
อาจารย์กอบพร I think your presentation, your slide is so beautiful, and it's your kindness to share with us. And how about you can share us the slide. Yeah. So in the name, no, yeah.
If someone sends us an email, we'll send you the slides. If you don't send us an email, we'll probably
No.
look at the slides.
Yeah, I mean
They were produced on behalf of the United States government, and anybody can have them.
I mean that I will upload your presentation in the website of the Virology Association. So anyone can go to the thaiviro.org and download it. Thank you very much.
All right, please come back around 10:45.